Thank you, Christine, and thank you everyone for coming. My name is Zili. I work at Guoping Feng's lab just across the street at the McGovern Institute. And yeah, Golden Neural Bloom. This image is part of the project where we develop gene editing therapy for Rett syndrome. So I'll probably start with a little bit of background knowledge about the Rett syndrome. So Rett syndrome is a severe neurodevelopmental disorder, meaning it happened in the brain and in the development. It's caused by mutations in the X-linked MECP2 gene. These mutations make the MECP2 protein either malfunctioning or non-functional. And because MECP2 is on the X chromosome, Rett syndrome predominantly affects females. And for biology experts who want the technical details, the patients are usually heterozygous for this mutation. And if we take a deeper look or a closer look at its genetics, 70% of these cases are C2T single nucleotide mutations. I know it's a jargon here, but to better visualize what I mean by a single nucleotide mutation, we can think about it like letters in a word, in a sentence. So DNA is written in letters, A, G, C, T's, and genes are like instructions written in their language. And I'll give some examples about, related to if one letter changes, what might happen to the meaning of the word. So the first example I can give here is we look at this sentence: I like the gray T-shirt, and then if we change only one letter, I like the gray T-shirt, basically the same meaning, except it's a little bit more British. This is in biology we call this silent mutation. So you change a letter, but it doesn't change too much. And then here's the second example: I got hit by a car, which is something I don't want. And if you change a letter, R to T, I got hit by a cat. Then you change some scary accident to something cute. And this is also one letter change, but you completely change the meaning of the sentence. This is what we call a missense mutation. And then there's a third situation in biology when you create a stop. If you change the step to stop, please stop, then the whole sentence just ends there. In biology, this is called a nonsense mutation, and the technical word is a premature stop codon. And Rett syndrome is related to the latter two situations. Missense mutations and nonsense mutations on the MECP2 gene causes the Rett syndrome. And so if we know that Rett syndrome is caused by only a single letter change, an obvious question that we can ask is, can we just simply fix that particular letter back to its correct state, so that it's like fixing a typo in a word, in a sentence, so everything will be fine. And this is actually what scientists are developing right now. There's a technique called base editing and a tool called base editor, which allow us to change only one DNA letter back to the correct one. And referring back to this example, I got hit by a cat, it's changed back to I got hit by a car. But there's an important caution here. In this example, we only want to change the T in cat to R in car. This is what we want. This is called on-target editing. But maybe somewhere else in the paragraph or in the article, you did another T to R, which you changed the time is tight to the time is right. This is what we call off-target editing, which is something we don't want. An important focus is trying to develop tools that are more on the on-target editing, but minimize the off-target editing. And for this purpose, we need a platform to look at where does this happen and how much does it happen, or does it actually happen. So Rett syndrome primarily affects neurons in the brain. So we need a system that is at least related to the brain. This is best we can have human neurons. And there's a little bit of technical details there, but we start from a cell state called iPSC, and then we step by step induce it to neurons, which we named it induced neurons. And in this state, we introduce our base editor and incubate for a while. Then we get our this state. And then we can use sequencing technologies to basically read out the whole, I guess, referring back to the analogy, read out the whole article to see if it changes any other words in other unwanted places. And this image is actually from this state when we incubate it with our base editor. So here, the golden color, really beautiful, is the map2, which shows you where the mature neurons are, our induced mature neurons are. And then the red is related to the last talk, so it's cell nucleus. It's has two importance here. It's where the DNA is at, and it's also where MECP2 protein is located. And the cyan color is our base editor expression. With that, I would like to thank the team. I think the whole team is here. Josh is an expert in microscopy, helped me a lot in taking this beautiful image. Shenjie is my mentor, actually the one in charge in this whole project, and Steven and our PI Guoping. And I would like to thank the funding sources as well from RSRT, Simons Foundation, Liangtan Collective. And yeah, that's it. Thank you.